So thank you so much for the introduction, and sorry for the quick technical difficulties with the slides. But I'm really excited to share with you all a little bit of the work I've been doing this summer as a graduate intern here. My personal background is largely in mammalian genetics, so this summer working with a lot of plant folks has been a really interesting and exciting opportunity. And one of the interesting features that both of these lineages share is complex multicellularity. And despite a lot of similarities in what these organisms look like, both during embryonic development and at some level in their cellular structures as adults, the most recent common ancestor of the plant and animal lineages is single cellular. And so this is convergent evolution of complex multicellularity. Despite this difference in underlying molecular logic or suggested difference in underlying molecular logic, there's a lot of similarities in how this complex multicellularity is established, including in the maintenance of stem and progenitor cell populations, which are active during the development of an organism, but also throughout the lifespan, provide support in tissue maintenance or repair and regeneration in response to injury. And these stem and progenitor cells are able to differentiate into specialized cell types, but are also able to self-renew. One tool for asking questions about multicellular cell types that's being used more and more is single cell approaches. And very rapidly, single cell atlases are being generated for a variety of species at a variety of different time points. So there's many across embryonic development, but also many in response to regeneration or injury. So I started this summer working on a set of 4 species. Arabidopsis thaliana is model organism, model plant, and then 3 close relatives that have much less cell type information known. So while this large body of single cell data sets are being generated, a major challenge is integrating or synthesizing the data that's being generated across different taxonomic distances, as well as using single cell data sets that are generated in non-model systems where the annotations for cell types or information about even sometimes the cell types that would be present in the data set are unknown. So a tool we were working on this summer to integrate data across species and to transfer labels from well-labeled to poorly labeled species is a tool called SAMMap. And so for these 4 species, we had data from roots, shoots, and seedlings, as well as really well annotated root and leaf atlases for Arabidopsis thaliana. And so using the tool SAMMap, we were attempting to put all of these single cell data sets that are in kind of their own gene spaces into a joint combined UMAP space. So the way that SAMMap does this is it initializes a graph based on sequence homology between the species that you're attempting to integrate. And so it starts out using sequence similarity between the gene sets of interest. And then it projects the data from your different species into that joint space. And then it relaxes this constraint on sequence similarity to allow for higher or lower degrees of connection between cell neighborhoods based on the co-expression of genes, even if they weren't really closely related in terms of sequence similarity. So this improves kind of the results that are seen with using one-to-one orthologs or just relying on sequence similarity, because it allows us to kind of discover other connections that exist between cell types. And so using this tool, we're able to combine these data sets into a single projection and connect neighborhoods of cells across species to transfer labels or to determine the types of things going on in different clusters of cells. So in this particular data set, not only are they ending up in the same space, we're seeing that cell and tissue identities are nicely maintained. So in the data set, we know that some of our single cell data sets were from shoot or from root or from whole seedling. And what we see on the right here, I think you might be able to see my cursor, is that in the upper right, we have kind of this section in the top that is mostly root cells. And instead of seeing each species in its own space in this graph, it's kind of like the TV static. Everything's really nicely integrated into one space. But roots kind of in one section of our UMAP, and shoots and leaves are in kind of a second section. And as you might expect, the seedling data is spread along this root and shoot sections of our UMAP. And while we're seeing nice integration of species, we're seeing some separation based on the tissue type, and then coarser or finer separation rather based on the specific cell types. So on the left here, these are the annotations we have from our really nicely high quality labeled root atlas for Arabidopsis thaliana, and you can see that even in this joint space, the cells that we know should be together are ending up in the same space. And in particular, this arrow I'm pointing to is the stem cell region of this UMAP space that includes sort of the stem cell niche and the quiescent center. So the other type of data we had in here, which is a little bit different from some of our other single cell data, was callus data. And calluses are regenerating plant tissue, so you kind of can cut it off and it's able to regrow, and sometimes a whole seedling. And so we were interested to see where this callus data was ending up. And what we saw is that to some extent, like the seedling, it's spread throughout the whole UMAP space, including the root and the leaf areas. But the vast majority of cells were ending up in this highly aligned to this meristematic stem cell-like region of the root atlas in particular. And so what we're seeing is that these regenerating callus cells transcriptionally resemble the root meristem, in particular the stem cell niche. So at some level, this suggests that the Arabidopsis regeneration process is resembling early developmental stages. So as it's regenerating, it looks like the initial generation. So this is really interesting. And part of why it's interesting is because this is very similar to how many animals are thought to do tissue regeneration. And so we thought about really pushing the limits of what we could do with the SAMMAP tool by integrating some other available single cell data sets of regenerating organisms. So in particular, we pulled in a zebrafish regenerating caudal fin and a fruit fly regenerating spinal disc to integrate into the same space as the root and the callus data from our plants. We've also, due to some computational and some summer limitations, the time length, we didn't fully integrate all of the other data sets we pulled, but we did pairwise run with the root atlas, a regenerating planarian regenerating its whole body, an axolotl regenerating a limb, a mouse regenerating a digit tip, and a human regenerating part of the liver. So we're able to get really good taxonomic breadth. But with the plants and with our fruit fly and zebrafish, we integrated and saw that the integration looked really nice. So we're still seeing kind of that TV static of all the species being pretty well integrated. So we're interested in seeing what types of cells were really closely connected in this space, because we do have nice annotations for these other model animal systems as well. And what we see is that our stem cell regions in particular are very well connected in the UMAP space and in our kind of alignment of transcriptomic identities. And so we see that for our joint embedding for our, I think, seven species, our fruit fly and zebrafish stem cells are largely ending up in similar regions to our plant stem cell niche. And then for our pairwise analyses, we also see that the stem and progenitor cells are ending up very highly aligned and often the highest alignment in the data set to the stem cell niche and the quiescent center for the roots. So if you look on the left here, this light blue here represents where the root stem cell niche and QC is. And that's where a lot of these cells are ending up for zebrafish and fruit fly. But on the zebrafish in particular, there's kind of the second region of stem cells. And that's because the zebrafish data set had actually two types of stem cells in its data set, the hematopoietic and the mesenchymal stem cells. And the hematopoietic stem cells are not necessarily as active in the regeneration of the caudal fin for the zebrafish. Instead, they're more involved in the production, like continuous production of blood products. So they're in kind of a more quiet state in this particular data set. So it's interesting to see that it does not end up in our stem cell region like our mesenchymal stem cells from this data set do. So that suggests that there might be differences in the stem cell states in how well they're aligning across species. We were interested in kind of seeing what tied this area together, what separated it from some of the other regions in our joint embedding space. And one thing that we saw is that this particular region is very highly enriched for orthologs. So genes that are not just sequence similar, but similar in descent and sort of evolutionary history. And orthologs are playing a pretty large role across our species here in connecting the stem cells. And in general, our quiescent center and stem cell niche had an outlier for the genes that are tying these regions together across species in the proportion of orthologs. But it's worth pointing out that still this is only representing about 30% of the genes that are connecting these cell types. So about 70% of the gene pairs here are not related by sequence orthology, even if they're very highly weighted. Looking still at the orthologs, we see that they're enriched for some processes that you might expect. Some very well-conserved processes. In particular, we are interested to see ribosome biogenesis, topic that's come up quite a bit today. And so we were looking at some of the specific gene pairs that were not only really closely tying these regions together, but also very highly expressed. And two particular genes we saw kind of across the board, not just in our multi-species embedding, but also in our pairwise embeddings, were the orthologs of NOP58 and NOP56, which in archaea is NOP5. And these genes are important for ribosomal function and substrate binding and release processes. In human cancers, actually, both of these genes are considered negative prognostic markers in quite a few different cancer subtypes, and are associated with increased stem cell phenotypes in cancer cells as well. So it does seem like they're playing a substantial role here in sort of modulating this pluripotent capacity of these cells. And it's thought to be involved in cell type differentiation by regulating our RNA modifications and through the production of lineage-specific mRNAs. So these are really compelling candidates that we're seeing, just that are very highly expressed, but there's a lot more pieces going on in this UMAP, lots of different genes that are connecting these different cell types. And there are, of course, many other cell types that we have not investigated quite as much. So there's a lot of open future questions coming from this summer, including how does this alignment change over regeneration time courses? Many of these atlases are generated sort of right after injury and then sort of along the regeneration pathway. So we're interested in seeing how alignment might change. We're interested in looking more in some of these gene pairs that don't have very high sequence similarity or not similar orthology, and interested in looking more at the function and structure of some of these genes to see how they might be playing a role in connecting these features. And we've looked largely at species with at least some regenerative capacity, and interested in looking, kind of expanding our taxonomic breadth more to look more at more limited species in terms of their regenerative capacities. And then what I'm particularly interested in is looking at stem cells at other time points, in particular the embryonic initial developmental time point, particularly if we think that part of what's happening in our regeneration atlases is that the process of regeneration is resembling initial embryonic patterning, whether we might be able to see higher alignments in the initial maintenance and creation of these particular cell type populations. So lots of other open questions and ways to use some of these exciting tools for integrating single cell data set, and lots still left to look at here. So we've used a lot of publicly available data here. So about 400,000 cells in total. I'm really grateful to all of the authors for making that available. Of course, I'm grateful to my mentor, Sharon, for all her support this summer and the others in the projects that I'm involved with. So at this point, I'm happy to take any questions.
Okay, do we have any questions from the audience? Okay, there are no questions, so I think for the sake of time, we'll go ahead and move on. Thank you again, Alexis, for your nice talk.
Thank you.